Hi guys, this lesson you are going to learn new terminology including isolation, gene pool, evolution, allele frequencies, Hardy-Weinberg equilibrium and Hardy-Weinberg equation, mutation, selection pressure, natural selection, and speciation. Let's get started. Good morning guys. The lesson today will be about the gene pool and genetic diversity. The learning objectives include first, understand how Hardy-Weinberg equation can be used to see whether a change in allele frequency is occurring in a population over time. Two, to understand that a change in allele frequency can come about as a result of mutation and natural selection. Let's review some definitions. Population, breeding group of individuals of the same species occupying a particular habitat and a particular niche. Habitat is a place or the environment where a plant or animal lives and grows. Niche of the organism is the functional role the organism plays within its ecosystem. Okay, the niche that we describe here, we call it ecological niche, which is determined by the habitat with its biotic or abiotic factors and comprises living organisms such as animals, plants, fungi, and other biotic factors, and its effect on other organisms. Population genetics is a branch of science or biology or ecology that study genetic variation within population and involves the examination and modeling of changes in frequencies of gene and alleles in population over space and time. In natural population, however, the genetic composition of a population gene pool may change over time. With the natural population, the main characteristic of the natural population is a change of allele frequency, gene frequency, and population gene pool are going to change over time or generation after generation. Gene pool concept. Gene pool is the sum total of all alleles in a population at a given time. All alleles of genes of all the members in the population in a given time, we call this gene pool. It's concerned with a single trait. Normally, when we say gene pool, we explain only one trait to focus on it, like a disease or advantageous characteristics or something like this. A population of organisms will have a particular gene pool with different alleles occurring with varying frequencies. So normally the population of the organisms that live in a certain area and interbreed together, they have a particular gene pool with different allele frequencies. This is what normally happens. We can define evolution as a permanent change in allele frequencies within a population. Allele frequencies features. In order to lead to evolution, the allele frequency should be not fixed. As the environment changes, the allele frequencies should change through the process of natural selection and adaptation. What is warfarin? Warfarin sodium is an anticoagulant medication. Anti means against. Coagulant means causing blood clotting. Warfarin controls the way the blood clots, thickens into a lump inside the blood vessels. This is why it can be used to treat the people who are susceptible for blood clotting. Now you are going to watch an animation explaining the history of warfarin and its evolution from rat poison to widespread clinical anticoagulant drug. Learn more about development of anticoagulant drug if you go to natural reviews. Cardiology interactive milestone. Let's get started. Rats and super rats. The brown rat is classed as a pest because it's going to consume some of the human food and considered to be transmissible to some diseases. People used to use warfarin to kill rats since the 1950s. Rat populations have been controlled by rodenticide called warfarin. The problem is the warfarin is an anticoagulant which acts by interfering with the way which vitamin K is used in the process of blood clotting. When food bait containing warfarin is eaten by rats, their blood fails to clot and they suffer from fatal hemorrhage, means excessive bleeding. As a result, most of the rats were dying when they got warfarin. But since the introduction of warfarin in the 1950s, population of warfarin-resistant rats have been identified in various parts of Britain, means that many rats have developed resistance against warfarin. Natural selection and warfarin resistance. Large population of rats compete for the available food. A random spontaneous mutation occurs within the rat population, conferring resistance to warfarin on one of the members. So at least one of the members of each collection or herd of rat will got warfarin-resistant allele. The warfarin-resistant rat is a selective advantage in areas where warfarin is used as a pest control. This means that the percentage or the frequency of the warfarin-resistant allele is going to increase in that particular population. The resistant rat is more likely to reach maturity, breed, and pass the resistant allele onto its offsprings. While the ones who are susceptible to warfarin, they are going to die and decrease in their allele frequency. In this example, natural selection is exerting its effect on a single major gene with two alleles. Some of the rats in the first generation are warfarin resistant and more likely to breed than the other susceptible brothers and sisters. So, after several generations or many generations, high proportion of rat population became resistant to warfarin as a mutant allele spread throughout the population. From this example, so we can consider that the poison acts as a powerful selective pressure or selection pressure and resulted in a rapid increase in the frequency of the resistant allele. Selection pressure is seen when a change occurs in an environment. Some individuals will have alleles which give them an advantage in the new condition, as when the rat poison containing warfarin is introduced to a habitat. Rats with alleles that give resistance to warfarin will be more likely to reproduce, increase in number, survive, and pass their resistant allele to the next generation. While the others who are susceptible to warfarin and have sensitive alleles, they are going to be discarded and removed from the population, and the frequency of the allele is going to decrease in the generations and the following generations. Eventually, the majority of rats became resistant to warfarin, and a new, more powerful poison had to be developed. Speciation involves a change in allele frequency. When the population and is driven by selection pressure over time, the number of individuals carrying a certain allele in a population determine the allele frequency. It describes that proportion of individuals carry a certain allele and is usually expressed as a decimal fraction of 1. The frequency of an allele in a population is not correlated within it, calls for a dominant or recessive phenotype. You will learn more about this in the next slide.
One of the world's most widely prescribed drugs. It's an anticoagulant which inhibits blood from clotting and is used to prevent and treat stroke, heart attacks, and deep vein thrombosis. So it's a pretty big deal. And it all started with some moldy hay and a load of dead cows. The story begins in the Midwest USA. During the 1920s, dairy farmers were losing cow after cow to a strange disease that caused their cattle to bleed to death. Vets soon identified the culprit, a crop in the cattle feed called sweet clover. If it was left to go moldy and fed to cows, it seemed to make them sick, but no one knew why. Unfortunately, few could afford alternative feeds, and so in the winter of 1933, one desperate farmer, Ed Carlson, made a 200-mile journey to the University of Wisconsin in search of help. In the back of his truck were 100 pounds of sweet clover and a milk can full of blood that wouldn't clot. Oh, and a dead cow. Eventually, he stumbled upon a biochemist called Carl Link and made a plea for help, although there was nothing Link could do for Carlson's cows. But this chance meeting would have a profound effect on Link, and he soon set his team on the hunt for the chemical responsible for the sweet clover disease. After 7 years of research, they found it, a compound called dicumarol which inhibited blood clotting in a number of animals such as mice and rabbits. In fact, one variant was so potent in rats that they developed it into a rat poison and called it Warfarin. Unfortunately for rats, it became one of the most popular rodenticides on the market. Soon, research showed that warfarin could be used safely in humans, but few doctors wanted to prescribe a rat poison. So, when it was approved for clinical use in 1954, it was sold under a different name, Coumadin. In 1955, it was used to treat President Dwight Eisenhower following a heart attack, which helped boost its popularity further. After all, if it was good enough for the most powerful men in the world, it was good enough for everyone else. Fast forward 50 years and other anticoagulant drugs have been developed, but warfarin is still used to treat millions of people. In fact, it remains on the World Health Organization's list of essential medicines. And all of this down to a chance meeting between a dairy farmer and a determined biochemist.
You'll watch a video now to learn how the warfarin works as an anticoagulant. Let's get started.
You may be taking warfarin if you're at risk of a stroke or have heart valve disease or atrial fibrillation, AF for short. Warfarin is an anticoagulant, which is known as a blood thinner. This is because it makes your blood less likely to clot and cause life-threatening blockages in your blood vessels, such as a stroke or a clot that has traveled to the lung. For a blood clot to form, you need vitamin K. This is produced by the liver and makes blood cells and platelets stick together and form a seal. Warfarin works by interfering with the production of vitamin K. This means blood cells don't stick together as quickly, reducing the risk of life-threatening clots forming. If you receive a hard blow or deep cut to your body, you may notice that you'll bruise more easily and that it will take longer for a cut to stop bleeding. Before you stop or change any medication, even if you're worried about side effects, talk to your GP first, as changing or missing a dose could harm your health. For more information about warfarin, visit bhf.org.uk/warfarin.
But how can we know how selection happens and how to find a value for the allele frequency? Weinberg have constructed an equation which helps you to calculate the allele frequency. According to Hardy-Weinberg principle, allele frequencies stay constant from generation to generation as long as certain conditions are met. Factors such as mutation, non-random mating, and selection can operate in natural population. These factors may lead to change in allele frequency and bring about adaptation and evolution. The principal force in bringing about changes in the genetic composition of population is that of natural selection. The allele frequency. Allele frequency describes the proportion of individuals carrying a certain allele, and it is usually expressed as a decimal of fraction of 1. The frequency of an allele in a population doesn't consider whether is it a recessive allele or a dominant one. Let's take an example. If we have two possible alleles, capital A and small a. If all the population there, we have 100 of the population, 100 individuals in the population, and suppose all of them are heterozygous, having the genotype capital A and small a. So we can say that the frequency of each of those alleles will be 100 over 200 or 0.5. This is just a model, but in reality we use a general formula that can be used to represent the allele frequency for the dominant and recessive phenotype. In the gene pool of the population, the frequency of the dominant allele is represented by capital P. So we use capital P to represent the frequency of the dominant allele and the Q to represent the frequency of the recessive allele. And we express that both of P and Q, if you submit them all, it will equal 1. Basically, we cannot use this equation to calculate the frequency or the difference between the heterozygous dominant and the homozygous based on the phenotype, because both of the heterozygous individuals and the homozygous dominant have the same phenotype. So this equation can be only useful to find the percentage or the frequency of the recessive allele in a recessive individual to the whole population. So we can know the distribution of the recessive phenotype in the population. And from this, we can calculate the genotype frequency using the Hardy-Weinberg equation, which it said that both of P plus Q equals 1. So this equation becomes useful. So it becomes useful because it can measure the distribution of the recessive allele in the population. Hardy-Weinberg equilibrium. Hardy-Weinberg equation, this equation, it's a general formula that is used to represent the allele frequency for the dominant and the recessive phenotype in the gene pool of the population, where P is the frequency of the dominant allele, while Q is the frequency of the recessive allele. Although this equation looks very simple, but it has an advantage because in any population it will be very easy for you to observe the distribution of the recessive phenotype, because the recessive phenotype is different from the dominant one. So it will be easy for you to distinguish the recessive phenotype in the population, and in this way you will be able to calculate the genotype frequency in the population. If you know the frequency of the recessive allele, so you can also calculate the frequency of the dominant one. Why it has a limitation? Because it's impossible to distinguish between the heterozygous and homozygous dominant based on the phenotype. So it does not show the difference between the heterozygous dominant and the homozygous dominant because both of them have the same phenotype. These two scientists, Hardy and Weinberg, they were working in two different areas in the world on the relation between the allele frequency and the change in the environment. Both of them have reached the same theory, so we call the theory Hardy-Weinberg theory. This theory is saying that in a population is not evolving. If the population is not evolving, the allele frequency in the population from one generation to the next remain the same, expecting that there is no any other evolutionary influences in the ecosystem. So if you have a population using the theory of Hardy-Weinberg, if the population Hardy-Weinberg give a zero value if the population is not changing or stable, and they give a value of 1 if the population is continually changing, or we can say it's evolving. The relation between the allele frequency and the stability of the environment have been studied and understood very well from Hardy-Weinberg equilibrium. Suppose you get a question like this in the exam. When a new species evolve, the gene flow reduces within the population, and you are asked to explain. This is very simply because breeding between the original species is no longer available. Hardy-Weinberg equation. Hardy-Weinberg equation, it has an advantage as it provides a simple model of theoretical stable population where P plus Q equals 1 to calculate allele and gene frequencies in population genetics. So we are able, by using the equation, to calculate allele frequencies and gene frequencies, to study and measure changes in species over time. So you can find the change in allele frequency generation after generation, so you can study the change in species or to know if the species is evolving or is stable. The algebraic equation can be expressed in that form. You know, P plus Q equal 1. By squaring both sides, we'll have P squared plus 2PQ plus Q squared equal 1. Remember, P squared is the frequency of the homozygous dominant genotype, Q squared the frequency of the homozygous recessive genotype, while PQ is the frequency of the heterozygous genotype. How to use the Hardy-Weinberg equation? You are going to count first in the exam, you are going to count first the recessive phenotypes, measure the frequency, which is Q squared, and then make a square root of Q squared. You will get the allele frequency, which equals Q. And then apply the Hardy-Weinberg equation, P plus Q equal 1, and then you will be able to estimate the genotype frequency. Let's take an example. Albino trait in humans. The dominant phenotype, if you have a dominant phenotype which has normal pigmentation, it has two possibilities of genotype, capital A, or capital A, small a. While the recessive phenotype has only one genotype, which is small a, small a. If you have 1 in every 20,000 in population is albino, so this value is the allele frequency. So it's a Q square. Okay, so this is the frequency, this is the genotype frequency, Q square. To find the allele frequency, you are going to square root this value. You will get 0.007. And then apply P plus Q equal 1. You will get P, which is the frequency of the dominant allele. 1 minus 0.007 equals 0.993. The conditions of Hardy-Weinberg equation, the population should be stable, and here the all relative frequencies P and Q, they should remain constant over time in the population, and also the frequencies of all genotypes P square, Q square, 2PQ, they should stay constant generation after generation. So we can summarize the conditions of Hardy-Weinberg equation in 5 important conditions. Number 1, no mutation is taking place within the population over time. Mating happens at random. Large population size, so no selection pressure, or no advantage of any types of selection, and isolated population. Isolated population means nothing enters or leaves the population. No migration, no immigration. 5, no selection pressures means that all genotypes will stay in the population and they are equally fertile and all of them are successful individuals. Bearing in mind that if you have large population size, you will ensure that the alleles will be randomly assorted in gametes, and therefore will have a large gene pool. Large gene pool means low chance of losing alleles. And this means that, for example, if we have a population of 10 individuals, only one of them carrying an allele Z, so we can say that only one individual out of the 10 individuals will have this allele. Suppose this individual is lost through predation or disease, this means that the whole allele will be lost from this generation and the successive generations, even if it codes for an advantageous trait. However, if the population has 500 individuals, so 500 will carry the advantageous allele, and the likelihood of all of those organisms being lost is very small. So there is a bigger chance of potentially useful allele being maintained in a larger population. This is one reason why large genetic diverse populations are needed to maintain biodiversity. So we can summarize here the importance of large population genetic through maintaining the biodiversity. How biodiversity can be maintained in large population? By first, random assortment of alleles, two, large gene pool, three, small chance of losing the allele, high probability of maintaining the potentially useful alleles. Isolation. According to Hardy-Weinberg equilibrium, isolation should take place in order to maintain the Hardy-Weinberg equilibrium. Isolation means no migration or immigration into or out of the population. But of course, this is not the norm. Here, the gene flow between the two populations is reduced. Selection pressure. According to Hardy-Weinberg equilibrium, in order to ensure equilibrium is taking place, all alleles would have the same level of reproductive advantage or disadvantage. This means that there should not be selection pressure. Also, we can say there should not be selection advantage for any of the alleles. No selective advantage for any of the alleles in the population. However, this is not always the case because the equilibrium cannot stand for a very long period of time. So normally the deviation happens from the Hardy-Weinberg equilibrium. This deviation shows that species are constantly changing. What are the factors that can bring about this kind of deviation? The most important factor is the selection pressure. Selection pressure means that some species or some phenotypes will be selected and they get a selective advantage over the other genotypes. Over time, this will lead to long-term change in the gene pool equilibrium, change in allele frequencies in the population. Finally, it will lead to speciation or formation of a new species. Mutations, or what we can say spontaneous mutations. Mutation is change that happen in the genetic material, so the allele are going to change or we can have a new allele. Mutations that happen in somatic cells are inheritable, means they will not pass into the successive generations. Only mutation that happen in the germline cells, we mean the precursor of the sperm and eggs, or we can call them spore mother cells. If mutation happen there, this will affect the allele in the next generation, not in the present generation, but on the next one. In plants, some changes that happen in the germline cells are not fixed. This is And this means that the somatic mutation that can happen in plant can be inheritable and go into the gametes into another part of the plant. So if mutation happen in somatic cells of a stem, you can see new features happen in flower or fruit of the same plant in the next generation. Most of the mutations are ineffective or the effective mutation happen very slowly or not rapidly. The great majority of mutations are recessive. If they are recessive, they will never be expressed, at least in the first or the second generation. Only the beneficial ones that will be expressed and will get established within the gene pool, and this will bring about evolution or speciation. What is the difference between random and non-random mating? Random mating means that the likelihood of two individuals mate is independent of their genetic makeup. So there is no choice for the male or the female for a certain phenotype or certain characteristics. This will ensure that the allele frequency will stay constant in the population. But if an organism is more attractive than another one, so it's more likelihood to have a non-random selection in this population. So when some features of phenotypes affect the probability of the two organisms meet, here non-random selection happen, or sorry, non-random mating happens. Non-random matings will lead after many generations to evolution through the natural selection, which is more likely to happen, more likely to pass the advantageous alleles to the successive generations. Finally, it will lead to speciation or evolution of new species. But what is the case in human population? Human population, the non-random mating is the norm. Here, gene pool will shift because different phenotypes that are desirable in some ethnic groups will affect the distribution of alleles or allele frequency. So if this process has been done for many generations, a shift in the gene pool frequency will take place. So a remarkable shift in allele frequency within the population will take place. And this is the case in human population. If you have a small population size, so we'll have a small gene pool. Small gene pool means high probability of the advantageous allele will get lost. So the genetic shift or drift would take place. Isolation. Remember that isolation was one of the points that should be maintained in a population in order to ensure hardy Weinberg genetic equilibrium. But if isolation is disturbed via migration or immigration, here the gene flow occurs in different population, means that one gene can flow from one population to another, and the organisms here are going to become more similar. We'll be having different organisms, different population, but have similar organisms. And this will ensure that constant change in allele frequency will take place within each population. Now, if isolation has been taken place and the mating between organisms in the two populations get reduced, the gene flow will get reduced, and here, when speciation is more likely to occur. Selection pressure. Some alleles code for the advantageous or disadvantageous phenotypes. So if you have a population or with some alleles that are different, some of them have an advantage, some of them have disadvantage, but all of them exist in the population. This will ensure the equilibrium, the Hardy-Weinberg equilibrium. But if the condition, if the environmental conditions have been changed, now a new selection pressure will be established. Some individuals would have an advantage over the others. These are the ones who are more likely to survive, and they are going to pass on their advantageous alleles or the successful alleles. Over time, genetic pool are going to change in genetic genetic makeup of the population and the allele frequency. So the natural selection now in progress or in action, and finally speciation is driven. Here is important terminology and their definitions that are needed for this lesson. Population, breeding group of individuals of the same species occupying a particular habitat and a particular niche. Gene pool is a sum total of all genes in population at a given time. Selection pressure, the effect of one or more environmental factor that determine whether an organism will be more or less successful at survival and reproducing. Selection pressure drives speciation. Hardy-Weinberg equilibrium, the mathematical relationship between frequencies of allele and genotype in a population. The equation used to describe the relationship can be used to work out stable allele frequency within a population. Gene flow, migration of either whole organism or genetic material into or out of the population and into another population, tending to make different populations more alike, but changing the allele frequencies within each individual population all the time. Now we are going to solve the checkpoint questions. Question 1. In a population of plants, 95 out of 200 individuals express homozygous recessive phenotypes for hairy leaves. Calculate the percentage of the population that is homozygous for the recessive phenotype, percentage of population that is heterozygous, frequency of homozygous for the dominant phenotype in the population. So in order to find out the percentage of recessive phenotype, remember that this value, 95 over 200, these are the number of recessive individuals in the population. So this value represents the Q square. Okay, so he's asking about the percentage. So just multiply this value into 100. Secondly, the percentage of population that are heterozygous. Heterozygous, to calculate it, I need the value of 2pq. Okay, so in order to get q, so you are going to square root this value. So q equal square root of 95 over 200. You will get the value of q. To get the value of p, you will apply this equation, p plus q equal 1. And as q equal, you will get the value of q from here. So 1 minus q equal p, you will get the value of p. Okay, this is the value of p and q. And then he is asking about the percentage of population that is heterozygous. So 2pq, you will get the value. C, frequency of homozygous for the dominant phenotype in the population. If you got the value of p, this value, just square it and multiply it by 100, you will get its percentage. Then question 2, gray fur in mice is recessive to brown fur in the population of 150 mice. 126 of them, 126 out of 150 have brown fur. Give the expected frequency of homozygous recessive, homozygous dominant, and heterozygous individuals in the population of mice and explain your working. So in order to answer this question, you are going to first find the ratio of the percentage of the brown fur in the population, 126 over 150, okay? Have brown fur, so the rest will have the gray fur, which is a recessive phenotype, which is 150 minus 126 equal 24. Okay, then find its percentage, 24 over 150 into 100. This will be the percentage of homozygous recessive individuals. This is 16%. This represents Q squared. Okay, So in order to get the frequency should divide by 100. So 16 over 100 will be equal 0.16. Okay, we said this is the value of q square. Okay, and then you will find the value of q by square rooting it. It's 0.4, and then apply the equation p plus q equal 1. Okay, so if you know q, so you can get the value of p. 1 minus 0.4 equals 0.6. Okay, remember this will be the frequency of the dominant allele. Okay, and this was the frequency of the recessive allele. Okay, and remember that both of them will be added to 1. 0.4 plus 0.6 equals 1. Frequency of the heterozygotes, which is 2pq. So 2 times p times q, you will get the frequency. The frequency of the homozygous dominant, you need to square root the value of p. Okay, so it will be 0.6 times 0.6 equal 0.36. Question 3. Explain why Hardy-Weinberg equilibrium is rarely observed in living organisms across the generation. Means why it's not applicable, why the equilibrium is disturbing all the time. So Hardy-Weinberg equation is not valid if it's applied to large population with a minimum of several thousands of individuals. The population must exist in isolation with no migration of organisms either into or out of the population. And all alleles must have the same level of reproductive advantage or disadvantage. These conditions are rare if ever met in a natural environment. Thank you guys. I will see you in the next video, inshallah.